Thank you, Rachel. That's a good point. So we will be recording every single session, and all the sessions, once they're recorded, they'll be converted into YouTube videos, and we will post them on the workshop page in the schedule. They'll be embedded in the schedule, and also you'll have access to them on our YouTube channel. Jesse.
I just wondering if you could clarify,
Mm-hmm.
you're recording the presentations, right? Not
Correct.
Individual breakout rooms.
Yeah, good point. So we're recording only the presentations, the main presentation in the main room. Thank you, Jesse. Okay, so on the homepage, I'm just going to go ahead and navigate to under the help menu. You will see the section here, learn how to use VEuPathDB. And when I go here, there are multiple options, including a calendar that shows you upcoming events. We try and keep this up to date. I can already see that there are a couple of events that are missing here, which we will add them. And so you'll notice here there's a section for workshops. And once you select this, it takes you to our workshop page, which includes all of our previous workshops with a lot of information on them. And here's the current workshop that we are in right now. If you click on this, it takes you to the workshop page, and down below there's a schedule. Okay. And so everybody should have gotten an email with a link to this, but if you forget where it is, just go to the help section and learn how to use VEuPathDB. And this is a nice place for you to go and look at and see if there are any new exercises in the future and things you want to learn about. It's quite useful. In terms of the workshop layout, you'll notice we have different sections. Some of them are lectures in the main room, like right now. And then there'll be sections that are in breakout rooms. And you'll see here, it says a breakout room on the section here. So depending on how many people are actually on, we will divide this up into breakout rooms. So we have at least one instructor per breakout room. The goal during the breakout room sessions is for you to try working with the resource, with the websites, and running through the exercises. So we have some very nice exercises prepared by various people on the project. And the goal is for you to walk through them step by step. They're very, very descriptive. They show you screenshots. And so you should be able to walk through them on your own. But during the breakout session, as you're walking through them, the instructor may demo some aspects of the exercise. So they will likely be sharing their screen. If you have questions, the instructor may ask you to share your screen to show everybody what you're having trouble with. Or you can just ask the question and then the instructor can demo a particular portion of that exercise. So I definitely recommend that you take advantage of this and really walk through the exercises, because that's the best way to learn how to really fully take full advantage of the resources. We do have breaks scheduled. So at 11 a.m., so in about 2 hours, I guess, we will have a half-an-hour break. So it's a good time for everybody to stretch, go grab a quick bite if you want. And then we'll get back and we'll have again some more lecture, and then followed by a few hands-on exercises. You'll notice that as you know, obviously from the information you received from Suzanne, our workshop is not a full day workshop because we realize that people have other responsibilities during the week, and also these get exhausting. And so this type of schedule will be repeated on a daily manner. So it's a mixture of lectures, which will be in the main room. So we'll come out of the main room, and then we'll be sent to breakout rooms for the various hands-on exercises. And Rachel, who is part of the VEuPathDB project, she's our Zoom MC, and she will be moving people around. If you have any questions or issues you're having with Zoom, or you somehow must move from your breakout room, definitely let Rachel know, and we'll figure things out. Okay, so anything else, Suzanne or others, do we want to say about the general workshop layout? And okay, great. Okay, so I'm going to spend some time right now talking about VEuPathDB and just making sure everybody is familiar with the basics. And so we're all on the same page. Again, I'm more than happy to be interrupted, so feel free to ask questions at any time. I will try and monitor the chat as well. So if you end up typing the question in the chat, I may or may not see it, but one of my co-instructors will definitely see it and either answer it in the chat or interrupt me and ask me to provide an answer, or someone else can provide an answer. Okay, so as Marianne mentioned, the VEuPathDB resources are funded through a mechanism called the Bioinformatic Resource Center mechanism funded by NIH. We also receive funding from the Wellcome Trust in the UK to support various aspects of the project, specifically FungiDB and TriTripDB. And also we have additional projects which we won't talk about during this workshop, but I'm highlighting here at the bottom of the page two particular projects, ClinEpiDB and MicrobiomeDB. So if you're interested in doing microbiota work or microbiome work, I would definitely recommend that you explore MicrobiomeDB. And if you work on clinical data, ClinEpiDB is also a fantastic resource for exploring various data coming from clinical epidemiologic studies all over the world. So those projects are funded mainly by the Bill and Melinda Gates Foundation. The ultimate goal of all of these resources is to make sure that you have access to the data and that you have tools and mechanisms to explore the data. And so VEuPathDB in particular has focused on mainly on genomic data, but also other associated data with the organisms that the genomes come from. And so a lot of the data is layered on these genomes. So functional data like RNA sequence or variation data or proteomics data or analysis of the genomes is layered on these genomes, and the genomes and the annotation on those genomes is used as a tether to then ask questions about the various underlying data. And our goal really is not to replicate what was in publications. Hopefully you can replicate some of the results that are in the publications by running the searches, but our goal is to provide the data in a way so you can ask your own questions about the data. So in effect, you're reusing results coming from various studies that have been published over the last 20 or so years. And we allow you to then ask questions about those results and integrate the results with each other. And I will illustrate this, and you'll be exploring this a lot during this workshop through the search strategy system. So I'm on the main VEuPathDB homepage. And again, if you weren't sure what VEuPathDB stands for, it stands for the Eukaryotic Pathogen Vector, Host, and Informatics Resources. So we support data from eukaryotic pathogens, the vectors that transmit these pathogens and other important diseases, and also the hosts like human and mouse and so forth that get infected by these various pathogens or get bitten by the vectors that transmit these pathogens. Okay, so the VEuPathDB homepage has a really hopefully straightforward layout. There's a top section, which is a banner section or header section, which includes the logo up here, so you know where you are, on which site you're on. I'll talk about the sites in a second. You have a section here at the top, which is the site search, and we have a whole exercise dedicated to site search. So this is almost like your Google search of the databases, where you can type questions. It's not as sophisticated as Google, but it's definitely sophisticated enough to allow you to find important genes and features. Below the search box, we have a number of menus which take you to different sections of the resource. So this header section it goes with you wherever you are on the resource. So these menus are accessible from wherever you are, which is nice. So you don't always have to go back to the home page to access the different searches or different help menus or different sections of the resource. So, before I go into these menus, I'm going to quickly mention this right-hand section here, which includes the various social media we're on. So if you're a social media person, you can follow us on Twitter and Facebook and Reddit and other mechanisms. And also we have a YouTube channel, which is linked right here. And so you can access these here. And also you'll notice here that it says my name over here, and that's because I'm logged in. And so logging in and creating a profile in VEuPathDB is very easy. And you can opt in to receive information from us or not. So that's totally up to you. But logging in is not required to use most of the site. There are some features on the site that require logging in because you have to set up your own work environment, or you're entering information that's linked to you. And we'll mention these throughout the workshop. But I highly recommend creating an account. You can do that right now as we're talking. If you go to the section right here and click on register, you can register. It'll send you a password automatically, and then you can log in. One really nice feature of logging in is that any searches you run while you're logged in will be saved in your profile. So you don't always have to come back and recreate and reinvent the wheel. And this way, it's a nice way. It's almost you're storing your lab book of your informatics searches, and then you can always come back to them and rerun them or examine them again. Okay, so coming back here to these menus, you'll notice here there's a My Strategies section. So if you're logged in, when you click on My Strategies, it'll take you to all of your searches, your previous searches, so you can explore these and see and go back and visit some old ones. So for example, I clicked on this in a separate window, and you'll notice here it's opening up a search strategy panel, something I've been running before probably. If I click on all, it shows you all of my search strategies that have been stored. Obviously, I use many of these in workshops, so I have many, but these accumulate for a while. There's no real limit on how many strategies you want to store. These strategies, when you save or store one of these strategies, you're actually storing the steps that you took in that strategy and not the actual results. So whenever you click on it, it reruns it. And so that's something to keep in mind, and hopefully will become more apparent to you throughout the workshop, that some of these results may change over time, right? So if there's a new annotation on a genome, obviously the gene models may change. And then as a result, if the gene models change, if there's RNA sequence data on that genome, the RNA-seq data may change as well, because we have to realign the RNA sequence data and determine expression levels for the new gene models, for example. And so those are things that are naturally would change, and your search strategy results may change as a result, okay? So that's one thing to keep in mind, and we'll come back to this, and hopefully it'll become more clear to you as we go on through the workshop. Okay, so coming back here to the home page, you'll notice here searches. So this includes all the searches that are available in the resource. This is the same section. You'll notice here it mirrors whatever's here on the left-hand section on the home page. So you don't always have to come to the home page to go back to run a search. You can just go to the searches and run them right here. And we'll discuss searches in a couple of minutes. There are tools. So there are a number of tools that are accessible and available through the resource. And we will talk about these throughout the workshop. I'll highlight a couple. Apollo is a tool that allows you to annotate and provide your own gene models based on your information. And the cool thing about this is that the changes that you make then become visible the next day on the resource, and somebody can go and see, oh, there's a community annotation, there's a community change that is available. And so that's very nice. And Uli will describe this further, I think tomorrow or the day after. We have BLAST, so you can blast obviously against any of the sequences. Let me go down here. Galaxy is a resource that allows you to run various bioinformatics tools on our genomes, and we will discuss this also in a couple of days. Tomorrow, maybe at the beginning of tomorrow. MapView is a tool to explore geospatial data, and so we will discuss this. And right now, it's available only for the vectors, but our goal is to make this available for other data types. But for now, it's on the vectors, and we'll get a nice description of this. A sequence retrieval tool. This is quite useful if you want to tailor the types of sequences you want to download and so forth. And we may get into it a bit more if there's a question, or describe it further later on. Yes.
Yeah, we've already had a question in the chat about whether we have a REST API or programmatic access to these. And I just wanted to point out that the last one in the tools menu is web services. If you need to access programmatically, that starts with instructions, and if you have trouble, you can always contact us.
Great, thank you, Suzanne. Okay, so my workspace, and we'll discuss this again in a couple of days, but also allows you to access results that you've analyzed in Galaxy, for example. And this is an expanding section, so hopefully over the next few releases, there'll be additional tools added to your workspace section where you can run various analyses. And there are other tools right here, or other items that belong to your workspace that you can access from here. Data, this includes information about the analysis methods that we use, the data sets that are available in VEuPathDB. I'll click to open up in a separate tab. We have data sets that we're working on, and also, the genomes or the organisms that are available in the resource. And so you can access all of these here. The data sets page, you'll see it right here. Right now, there are over almost 2,800 data sets available in VEuPathDB. And so you can go in and search them. You can filter them on different aspects, like RNA seq, for example. This will filter. And so now you'll see these are all the RNA seq data sets in the resource, or at least that have the word RNA seq in them. And so the different ways you can filter these, you can navigate them and filter on specific aspects. The bottom line is that there's a place for you to find and access all the data sets that are available in the resource, and ways for you to search them. About has a lot of information about the resource. So if you're curious, well, what is VEuPathDB? What is our mission? What are our goals? If you're looking for any of our logos, I highly recommend, if you use the resources, first of all, to cite us in your papers. That's super important, not only for transparency's sake, to let people know what resources you're using so they can replicate your work, but also important for us, so there's awareness about these resources, and funders in particular are aware that these resources are useful, are being used. And I would highly recommend that you include one of these logos if you use them, if you use one of these resources in your presentations, in your acknowledgement slide. So just like you acknowledge somebody for giving you a reagent, it's always nice to acknowledge the resources where you did your in silico work, so that the audience is aware of this. There's a lot of other information. If you're interested in submitting data to us, we have a whole section on how to submit data to us, and with a lot of information. So I'd recommend that you access this. If you're interested in, well, what kind of papers cite us, you can go here and learn more about how, well, you can learn how to cite VEuPathDB publications. What I meant to show you is you can see the papers that are either papers that are that we've written that describe the resources. So if you're wondering, well, is there a publication that describes VEuPathDB? Because either I would like to put it as a citation in my paper, or you want to read it so you can learn more about the resource, and you can go to that section, and you'll see here that there's a nucleic acid research paper recently published, which you can read, and it's freely accessible. If you're interested in knowing, well, what publications out there cite the resources, you can click on this. This takes you to a Google Scholar citation. And then as you scroll down, you can start seeing the various publications that are out there that cite our resources, and you can explore them and see them. So this is nice. And so you can obviously see, since in 2022, there have already been 1,600 publications. So they're heavily used, and people do mention the resources, but super important to do this for continued funding, and also to keep for transparency's sake. Help. So if you ever have questions about the resource, you're doing something and you're just not sure how to do it, I highly recommend going to the Learn How to Use VEuPathDB section. So in addition to the workshops, which include the detailed exercises, we have an FAQ section that we try and keep up to date. So if we notice questions that are coming to us on a regular basis, we update this section, and it has information with graphics. And so I would definitely recommend that you come here and take a look at this section and see if you find an answer to the question that you have, if it was a more general question. But if you don't find the answer, even if it's here and you're just not sure what it means, I highly recommend that you just drop us an email. And the best way to do this is to click on the Contact Us link. This is super easy. You just go in here. If you're logged in, your email is auto-populated, and just type your question in here, send it to us, and many of the instructors on this Zoom session will receive the email, and somebody will respond to you. We try and do it within 48 hours. Sometimes it takes a bit longer, but our goal is to respond to all of our emails and make sure you get an answer. This is heavily used, and we love to hear from you. And you don't have to only send us questions about something you're not sure about, but also if you notice a bug on the resource or something that's not working right, just let us know. Even if you're not sure if this is a bug or not, it's good for us to double check things. It's a really big resource, lots of sections, and so having multiple eyes on it is quite useful. And so anytime you notice something that is not working, just let us know, and we'll try and fix it or find a solution for you. Okay, so that's as far as the menus at the top. Before I go into the searches, just quickly here, the central portion is really a help section. It's informational. You have some tabs here that give you some information about the various tools and different data. And you can read more about the different data types. So if you're interested in, well, how do I explore phenotypic data? This gives you some example of phenotypic data that are available in the resource and how to explore them. If you're really interested in the genome browser, you can click here. It'll give you some information on how to access the genome browser. Search strategies, again, same thing. So all of these are informational. There's a generic introduction to the resources. So if anything I said today is not clear, even if it's not clear in the recording, you may want to explore some of our videos on YouTube, including introduction, which may give you an answer. On the right-hand section here, there's a news and tweet section. So if you click on this, this gives you the news. So we just released release 59 late last week. And so if you're curious, well, what is new in release 59? If you look in the news section, either you can scroll in here. If you're only interested in looking at ToxoDB, for example, you can click on this, and this will take you to the news section for what's new. And well, this is taking you to the full news section. Then you can scroll down and look at the various resources and see what's new in those resources, both in terms of features and tools. So one thing that's really cool about the VEuPathDB resources is that they're all built on the same infrastructure. So anytime we develop a tool for one of the resources, assuming the data is available in one of the other ones, all of them will get the same tool. And so that's pretty cool. It makes it a bit easier to manage the different resources. And I keep mentioning different resources. So VEuPathDB has various component sites, largely for historic reasons and maybe where funding is coming from and so forth. But you'll notice here these logos on the bottom. And if you look at the About section, VEuPathDB sites, here's a listing of them and you can access them. And really the only difference between these, I'm just going to open a bunch of these in the background. The really only difference between them is the color of the resource. The layout is the same, and the data content, right? So if there's no RNA sequence for Acanthamoeba, for example, which is an AmoebaDB, then there won't be any RNA sequence for Acanthamoeba. Or if there's no phenotypic data for any of the amoeba, whole genome phenotypic data, then there won't be a phenotypic search in the resource, which obviously makes sense. But as I scroll here through the different resources, you will notice that they all pretty much look the same. And the only difference is the color. And of course, when you delve into the searches, you will start seeing some differences. Okay, in terms of the searches, I'm actually gonna use maybe PlasmoDB as an example, because it has a variety of searches here. You can expand all, right? And you can start scrolling through them and look for the type of search that you're looking for, that you're interested in. or you can filter the searches by typing a keyword at the top here. So when I come to one of these resources, I'm usually coming with a question, right? I'm interested in discovering something new using the type of data that's available. And so, for example, if I'm a Plasmodium researcher and I'm interested in finding genes that are expressed at a particular developmental stage of the parasite, I may be interested in looking at, well, what RNA sequence data sets are available in the resource that I can start using to explore. And so up here, I can either look for the search and try and figure out, well, where is this search found? Or I can just start typing the word RNA. And as you see that now, all of a sudden, things are filtered to where the word RNA is appearing somewhere. And you'll notice here that RNA appeared in multiple sections, but what I'm interested in is a transcriptomic section. You'll notice we have microarray data, RNA sequence data, and single cell RNA sequence data. I'm going to go ahead and take a look at the RNA sequence evidence data. And this now gives you a list of all the available searches that are RNA sequence data sets. You'll notice that there are different organisms. So Plasmodium berghei, for example, Shabauti, Plasmodium falciparum. The data set name, hopefully it's descriptive enough to give you information about what kind of data it is, or maybe what kind of developmental stage it is. You have information here about the experiment, and also typically the first author of the publication or first authors of the publication. If this is not enough information for you, then you can mouse over this little question mark next to each of these questions. It gives you a pop-up window with a little bit more of a description here, a summary, and a link to the actual publication if the data is published. And so this helps you then explore the data set itself. And I'll show you in a second where you can access our data set page for this particular data set. To the right of the search, you will notice there are different boxes here. Each one of these boxes denotes a different type of search that can be run against the data from this experiment. And you'll notice there's a key at the top here that tells you what these are. Typically, the main ones that people are interested in are the differential expression, fold change, or percentile. And we'll discuss these in a bit more detail when we get to those searches, but very quickly, differential expression is when you have biological replicates, so you can use a p-value. Fold change is simply looking at the difference between different samples based on the fold change expression between them. And percentile, you're looking for amount of expression compared to all the other genes in a particular experiment. So if you're saying, well, I want to find all genes that are expressed at the 90th percentile or above. So you're looking for genes that are super high expressed compared to the rest of the genes in the genome. Okay, so let's say I come here and I'm starting with my question. I'm interested in finding genes that are expressed at a particular developmental stage. So let's say I'm interested in genes that are expressed in gametocytes. So I can start typing the word gametocyte up here. And now it filters the searches. Hopefully, if these are annotated properly, it will return experiments that have gametocyte stages in them. And so I can quickly look at this and then decide, well, which experiment I want to start searching first. And so I'm just going to randomly pick one. I'll just go ahead with this Plasmodium berghei, female and male gametocytes. Let's say I'm interested in, well, as I'm looking at this, I may say, oh, actually, I really want to, determine if there's a difference in genes that are expressed between males and females. So I'll go ahead and run my differential expression search. These search pages every time you come to a search page on any of these resources, they may get super complicated or look super complicated, but usually there's a lot of help available. And if you scroll through it slowly, it's hopefully fairly intuitive. I'm on the search page. If you scroll down to the bottom, there's usually a little bit more information about the search itself. There's also a link here that takes you to the publication again, but also to the data set page for this particular experiment. So I open this up in a separate tab, and you'll notice here now there's this experiment, females and male gametocytes. This is the link to the publication, whose lab this came from. As you scroll down here, you'll see that there's a nice description. It's not always as nice as this. This one is a particularly very nice one, and but I picked it by random, so that's good. As you scroll down, you'll be a link to all the searches that are available, and if available, we have the link to the repository where this data came from and an example gene that has expression from this this particular experiment. In this case, this gene is super highly expressed in female gametocytes, at least differentially expressed compared to the other stages. Okay, so back to the search, and up here. And so let's say I'm interested in finding genes that are upregulated in females compared to males. I'm going to pick the sense RNA sequence data because I'm interested in the sense expression. You could obviously look for genes that have antisense expression, but we're not going to focus on that right now. And let's say I'm interested in genes that are upregulated, so I can change the direction of regulation. And then I will go ahead and change, let's say, the fold change to 4. And let's make this a 0.05, 5 times 10 to the minus 2. Okay. And so now when you get answer, it returns 788 genes that met this criteria. Okay. And so you may decide that, oh, this is great. I'm satisfied. I found, my genes that are upregulated in males. Let's see, what did I choose? In females compared to males. And so you may be satisfied and be done with this. Or let me do this one more time. So what I did is I came here to the step in a strategy. So as soon as you run a search, or you come from site search and you export it as a strategy, what happens is you start up with what we call a search strategy. It's basically an in silico experiment. We're in effect running an experiment here. We asked the question about an RNA sequence data that's available. We asked for genes that are upregulated in female gametocytes compared to male gametocytes. And it gave me the results, 788 genes. This is the beginning of my experiment. So the analogy I like to use, if you work in a lab, it's just like running an experiment in the lab. You typically have multiple steps. If I'm doing something simple like an enzymatic digest, you may start with your DNA and your enzyme. You mix them together. You might add a buffer in there. And then that's your first step, let's say. And then you take that and you put it into the next step. Maybe it's an incubation at 37 degrees for an hour, okay? And then you take that and you may now purify your DNA, okay? So that's a workflow, and it's an experiment, right? It has multiple steps. Same thing here. Your experiment, this is in silico, it's on the computer, and it can be composed of multiple steps, or it can end here depending on the question you're asking. So once I click on edit, when you mouse over this, you'll get this little edit icon right here. I can click on this. It shows you a pop-up which gives you information about the parameters you chose. So this is good because I sometimes, once I do this, I forget, well, what did I actually pick? Females compared to males? Did I compare males to females? And so it's always good to double check this. But also it offers you a bunch of additional things that you can do with this step. And so, for example, I may decide, well, 788 genes, that's a little bit more than I wanted. I really want to find things maybe that are even more highly regulated, upregulated. So I can revise this step. And you can do this with any search that you run that comes in your search strategy, you can revise it. And so now let's say we're going to change this to upregulated by tenfold. Okay, we're going to be a bit more stringent here. So you still have a lot of genes, so 447 genes that are upregulated by tenfold. That's totally fine. And so now you can explore these genes in different ways. So one way is you can very quickly, I'm looking at the product description, I could look at the names, and I can try and see if I find a pattern here of names. Of course, that's very hard when you have hundreds of genes. One thing you can do quick and dirty is you can generate a word cloud that's based on the words that are present in the product descriptions. Of course, you'll find things like the word putative or function, which are not, or protein, which are not terribly useful, but you can actually filter these out of your word cloud, and this will, at some point, give you something meaningful that you can potentially use. And so let's do a couple more. And so now if you mouse over any of these, you'll see that membrane appeared 21 times. So membrane proteins are in there. kinase appeared 14 times, and glidosome appears 6 times. So those are interesting words that are appearing in your results. You may, you can change the structure of this word cloud. Sometimes it makes it easier to see things. Okay. The other thing that you can do is, which is more statistical, is you can analyze your results and run a Go enrichment on your results. Okay. We will do this in a different day on the workshop, so I'm not going to go through the process of running this, but just to let you know that you can actually do this and get something more statistically robust. Okay. So let's say I'm not satisfied, I'm not done. I really want to carry this further, and I want to know if any of these genes that are upregulated in female gametocytes compared to male gametocytes are also secreted. Okay, so remember, you're the scientist here. You're coming in with the question, and your question could be very sophisticated. You can start off by saying, I want to find female gametocyte genes or proteins that are upregulated, that are secreted, that potentially have polymorphisms and or have a phenotype, maybe are essential, and so forth, right? And so you can really build a complex question. And so I'm going to add a step. And so let's say we want to find things that are secreted. So I'll go ahead and add a step. And when you click on Add Step, you get this pop-up window, which is offering you multiple things. But I'm going to combine my previous search with another search by genes. I'm sort of searching in the gene world right now, because these are data sets that are mapped to the genomes and the genes. And I can choose how I'm going to combine the results from my previous search, which is the female and male gametocyte search, with the new search that I'm going to run, which I'm going to select from the menu of searches here for secreted proteins. And so I can start typing the word secreted in here, and you'll see now there's a search called predicted signal peptide. And so I can click on predicted signal peptide here, and I can, in my case, I was working on Plasmodium berghei. I mean, honestly, because I'm using an intersect, you can select all and not even worry about it, and the filtering will do it for you, or the intersect will do it for you. So I'm going to run this step. So what happens now, I'm asking for any gene that has a predicted secretory signal peptide in the database. There are 34,000 genes that met the criteria. Out of those, 84 genes also were upregulated in my female gametocytes. Okay, so now we have something a bit more maybe relevant to the question that I'm interested in. So if you're interested in finding genes that are secreted by the female gametocytes that are somehow maybe involved in survival, or maybe important for after they're taken up by mosquitoes, or maybe they're things that you want to develop a new vaccine against, and so forth. So there are lots of things you can ask about this. I'm going to highlight a couple more quick things. One is that note here that I'm in the Plasmodium berghei world, right? But let's say really you're working on Plasmodium falciparum. You want to work on the human malaria, and so you're interested in knowing, well, are there any genes, homologs of these genes in Plasmodium falciparum? So one way you can do this, obviously, you can download all the sequences and blast them against Plasmodium falciparum genome, figure out which ones are the best blast hits, and go take that route. The other option is we already compute the orthology or the homology of these genes using a resource called OrthoMCL, and we will discuss this as well on a different day when we discuss the orthology. But we have a way for you to quickly transform these results from berghei into falciparum, or into any other organism for that matter. And so instead of now combining with other results, I'm going to transform into related records. And when you click on this, you'll have different options here. One of them is ortholog. So I'm going to click orthologs. And in my case, I'm interested in falciparum, and I'll pick the reference, which is Plasmodium falciparum 3D7, and we can run the search. And so now what happens, it tells me that these 84 genes had 79 homologs in Plasmodium falciparum 3D7. Okay, so now you're working in the Plasmodium falciparum world, which is nice. The other thing you can do is now you can add additional experiments from Plasmodium falciparum in this case. So I can start looking in here. We said maybe we're interested in genes that are essential based on a phenotypic experiment, and so we can see if we have phenotype. And again, don't worry about remembering the specifics of every search I'm running. I think the point here is that you can get very sophisticated with these search strategies and really develop something that's a multi-step search strategy that that hopefully gives you some answers to your question. And remember, since you're not wasting any lab reagents, really, it's just your time. There's no wrong answer really. Even if you get an answer that's 0, well, at least you can go back and modify the steps, and you haven't wasted all your DNA or your enzyme. So I'm going to go ahead and select this. Again, I won't go into the details of the study, but obviously you can do that. If you scroll to the bottom of this, you'll see here the links to all the papers, and also we have information on our dataset page as well. And so there's a lot of help, hopefully, for you that you can go back and learn more about these studies. But in my case, I'm going to look at the mutagenesis fitness score, and I'm going to look for any gene that has a mutagenesis fitness score that's below -3. So I know from the study that these are potentially genes that are really important for fitness. You may go below -2, but I'm going to pick -3. And so now I'm going to run the search. And what this told me is that there are 1,109 genes that had a mutagenesis fitness score based on that met the criteria that I selected. And 10 of those were in the 79 genes that are orthologs of the potentially secreted female, upregulated female gametocyte genes. Okay, so now we have 10 genes that are potentially secreted that may be important in gametogenesis, or at least are expressed in gametocytes and may be important for that or for other stages. So you may wonder, well, what's going on here? This gene is called liver merozoite formation protein. Okay, why is it expressed here? So just like with any experiment, if you're running a gel and you have controls on your gel, you're going to look for controls in your experiment here. And so hopefully you will find some genes that are known to be secreted in female gametocytes. And sometimes you'll find genes that are false positives, that shouldn't be there. And that's okay to find those things. But one note about the gene names, just because it's called liver merozoite formation protein, it doesn't mean that the only place it's expressed is in liver merozoites. It may be expressed in other stages. It's just that when the name was given to this protein, that's all that was known about. Of course, it may be known that this gene is specific to liver merozoites and should not be expressed here. And then maybe you should go back and say, well, maybe I did something wrong, or maybe this experiment is not great in pulling out female gametocyte genes, right? And that's totally okay, right? So you should not only question how you set up the experiment, but also question the data where those experiments are coming from, where the results are coming from, right? These papers get published, they're reviewed, but as we all know, I've read many papers out there, you have, I'm sure, as well, where you look at the results and like, yeah, I don't think I trust these results. There's something really fishy about this, or I don't like the conditions they use in this experiment, or maybe their female gametocytes were not really all female. There were some males mixed in there, or so forth. And so you should go through the same sort of thought process as you're looking at these results. Just because you're getting a list of genes, it doesn't mean they're 100% accurate. You should then, as a scientist, you should look at them and make some conclusions about them. Okay, so this is a search strategy. We'll be using it a lot today and learning how to use it, and hopefully by the end of today, you'll be very confident in how to build a search strategy. We can't give you the questions that you're interested in. You have to come up with those questions, but that's obviously something that you either have or you will develop to know how to put them together. Okay, so very quickly, any one of these genes, as you can see here in this list of results, is linked to a gene record page. So that's imagine a Wikipedia page for the gene, basically. So it has all information about a particular gene that we have. And so let's go ahead and take a look at this liver merozoite gene. I'm curious to see what's in there. So let's take that. And so what happens here is that you start off with this gene page. At the top of the gene page, you'll notice there's the gene ID. So this is a unique identifier that you can use to put in your paper or search for it. And it should be consistent. And if gene IDs change, we do our best to map them. So even old gene IDs from 20 years ago on the initial Plasmodium annotation project can still be searched within the resource if they have a mapping. You'll see here, this is the product description of the gene. So it's a liver merozoid formation protein. It's also called PALM, P-A-L-M. As you go down, it's protein coding. You can see it's on chromosome 6. This is the exact coordinates of this gene. It comes from the strain 3D7. This is a particularly curated strain. We have curators on our staff, and they over time they add new annotation to these genomes. So genomes that are curated get a little bit of special attention from a person. As you go down here, you can add a user comment on this gene. So if you're logged in, this is a way for you to add, if you know something about this gene. So let's say this gene is called liver merozoid formation protein, and you know that actually, no, this is also a merozoid female gametocyte protein that's super important for, infection or for take up by the mosquito or for survival within mammalian cells before they get taken up. Then you can actually add this information on the gene page. And when you click on add first comment, again, this is one of these features that requires you to log in. So I'll go ahead and click on log in quickly and go through the motions of logging in. Okay, and so now you get this section where you can add a user comment, and it's fairly straightforward. You just add the information here, submit it, and as soon as you submit the information, your comment becomes live on the gene page. Okay, and so that's pretty cool, right? So whatever you change, whatever you add, whatever comment you add, it becomes live on the gene page. And if we have time at some point, I will demo some of these. Okay, I'm not going to go through the full details of the gene page. Suffice it to say that it's made up of many sections, and you will see here this table, which is a table of contents basically for your gene page. You can expand all. You'll see there are all these subsections, but also you can filter. So I can quickly, for example, say, well, I'm interested in looking at the phenotype data, and so I can start typing phenotype, and let's go ahead and take a look at the phenotype graph. And as you come in here to this section, the phenotype graph section is right here. I had searched the piggyback data, and then you'll get a graph here that shows up for this gene. And so my fitness score that I selected was down here, below -3, and so it is right there. So that's good. It meets the criteria, which is good. You can scroll down and look at the transcriptomic data. So remember, when I searched the data, I searched the data from female and male gametocytes, but those were in Plasmodium berghei. And I'm looking at the Plasmodium falciparum gene. So the berghei data is not going to be available here, but I can quickly see, well, is there any gametocyte data available for falciparum? And so you can start looking here and seeing if you find an experiment that's worth looking at. So here's this one here. It looks like it's a fairly detailed experiment. And you notice here, the type of experiment, this is not not an RNA sequence data set, this is a microarray data set. So the dynamic range is going to be different, obviously, but as you look here, it looks like this is increasing over time, the expression at least, and then goes down. It's not increasing a lot, but it is there. We can look at this experiment here. Here's another one. This one has females and males. So let's see what this experiment says in Plasmodium falciparum. And so this is kind of exciting, right? So this is an experiment from a totally different lab on a totally different species of Plasmodium. They compared females and males, and in falciparum, the females are also upregulated compared to the males, right? And so that's pretty cool. So my level of confidence in this gene being upregulated in female gametocytes has just increased, right? Because now I found another experiment that supported this criteria. And the same thing, you can go in through all of these and try and determine for yourself if any of these make sense. And here again, this is not distinguishing females and males, but you can see in the gametocyte stage 5, they're much higher expressed than the other stages for whatever it's worth. Okay, so there's a lot of, the point here is that there's a lot of data you can explore in the database and worth looking at. One thing to point out here, I noticed as I was scrolling down, this gene does not have a signal peptide. So remember when I ran my search, if I go back to my search strategy, I ran my signal peptide search on the Plasmodium berghei genes. But this gene that's a falciparum gene, that's a homolog of one of these berghei genes, actually does not have a single peptide based on the prediction. And so this may be, there may be multiple reasons for this. One reason could be that the gene model in Plasmodium berghei is incorrect. And really, the beginning of the gene is not the right beginning. And if you modify the gene model, it actually does not have a single peptide. The other possibility is that the gene model in Plasmodium falciparum is incorrect. And in fact, the gene model should be extended a little bit, and you then will be able to figure out what is that actually you'll find that it has a single peptide. We have an exercise where we're going to talk about gene models as well, so we won't talk about it right now, but the data is available for you. For most of our organisms, or related organisms, there is data in there for you to make your own conclusions. And so, so you should never just trust the official annotation if there's RNA sequence data available in a database, because you can go in and look for yourself and decide whether you trust the gene model or you think the gene model should be modified. And this may be an example of one of these cases. Obviously, another possibility, there's a true biological difference between Plasmodium berghei and Plasmodium falciparum, and this particular gene is secreted in berghei and not secreted in falciparum. That would be a possibility. We can't rule it out. And I'm sure there are other possibilities that could cause these differences. Okay, so I will maybe show you one more thing, Suzanne, and then we can switch over to the next section. I believe I have until now, basically, right?
Yeah, I think you're good. You're 2 minutes over, but we'll give you another 1.5.
Okay.
Yeah,
Perfect.
do what you need to do.
So just very quickly, I wanted to point out that in addition, so we talked about the search strategy system, right, that allows you to do searches. We will exercise site search in a few seconds now in the first exercises, as well as search strategies. And then the other place where you can explore the data is in our genome browser. For any gene that you go to, and I'll just pick another one here. For any gene that you go to, we have an integrated genome browser that allows you to explore the data in a visual way, sort of walking across the genome. And you can access the genome browser from different places. So one place is right here on the gene page. And so you can go down to the gene model. I can see here, oh, this gene is a multi-exon gene. I know this because, let me zoom in a little bit. I know this because these blocks here are exons. So these red blocks are exons, and in between there are the introns. You'll notice there's some red genes and some blue genes. This is just the direction of transcription. And so remember that DNA is double-stranded, and so a gene could be on one strand or the other. And so this is showing you the direction of the transcription. And so here's the gene I'm interested in. It has 1, 2, 3, 4, 5 exons, it looks like. Let me zoom out again. And if I want to explore this gene further, I can click on the genome browser. I'll let it open in the background here, and it'll open a genome browser which you can explore further. We also have the genome browser view embedded in different sections, depending on what you're interested in. And so we have down here, you can look at the protein features, for example. And so, for example, this gene looks like it has a specific domain that's a Plasmodium host traversal domain. And so that's, again, this, I switched, I'm not looking at the liver protein, but I'm looking at this sporozoite host traversal gene. So that's could be quite interesting, obviously, again, from a biological standpoint. This particular gene does have a single peptide in falciparum, which would make sense if it's a host traversal protein. And then, as I mentioned, I opened up the genome browser in a different view. So here's the genome browser. When you click on it from a gene page, it takes you directly to this gene. It's highlighted right here. And again, we will have a whole exercise on the genome browser. You can zoom in, You can visualize different tracks, and I'll just click on this. I won't play around with it, but there are hundreds of tracks, data tracks that you can load. Of course, this is bewildering and could be daunting, but again, the goal is that if you're coming in with a specific question, then you start thinking, well, what kind of data can help me answer the question that I have? And one of them may be going to the genome browser and looking at RNA sequence evidence to explore the gene model, or looking at ChIP-seq data because you're interested in determining where's the true promoter for this gene. And so there are multiple different types of data sets that are available for you to access. Okay, I think with that, we are right here right now, and I think we are the first sort of where it goes back to your hands is going to be our hands-on exercises. So I will stop talking here, and Suzanne, I don't know if you was anything you want to say before we switch into our breakout rooms.
Sure. So we have 3 exercises for you here, and they're the site search will define how a lot of people enter our sites and look for gene IDs and certain things. The organism preferences is something you want to use to possibly help your searches run faster. You can choose a subset of organisms within each site to search. But you have to be a little careful because the menus also are subsetted at that point too. So if you choose organisms that don't have a particular type of data, then that search will disappear. And the gene pages are really just full of data and information. So this exercise takes you through the basics of looking at and navigating the data. And as you go through that, you might want to think about what types of data you'll be using. And then the scheduled links to each of these, and you can just follow each of those directions with your instructor. Yep. Breakout rooms, I think, is what we need.
Yeah,
Rachel,
I think Rachel, I don't know if you determined how many breakout rooms you want to put out, but I think we have sufficient instructors to have a good number of breakout rooms and a good ratio of instructor to student.
Right, she has 9 rooms ready to go.
Okay, perfect.
All right, and I'll open those now.
Great, thank you.